Yeah, well, I think it's over there. Okay, guys. Hi, this is Ian. I can't pronounce his last name. It's something too good, too good. Something complicated. It's too easy, but it's complicated. Ian is coming from Edinburgh University. He is working on cerebellum, motor cortex, and what's in between. In relation to the things that Art was talking to you last week. So he's going to give us two talks. One today, which is kind of more introduction to the cerebellum, and then on Thursday morning it will be a little bit more advanced talk on what's going on now in cerebellum research. And actually he has a busy schedule because Thursday evening he is also giving the LSIC seminar, and this time would be on motor cortex.
You really are getting your money's worth. You really are getting your money's worth. There's actually the two lectures I give here is almost the same as the years worth of lectures that I give in Edinburgh.
How did you manage to do that?
Because I'm a welcome fellow, so I don't actually have to teach back home. So you've actually asked me to do as much lectures as Edinburgh University for an entire year. Go figure. Just because you're a friend, that's taking advantage.
Exactly. Anyway, yeah, we'll sort it out between us. Paying in a different way. So yeah, enjoy the talk. It's a brilliant talk.
Before you start, can you turn the microphone?
Yeah, sure. Are you doing the lights on the front here?
I think it's already set up, the microphone.
No. Dim the light. That'll do. Okay, is that better? Good. Okay, so yeah, as Mickey said, this lecture is going to be almost a purely didactic lecture on the cerebellum, everything in it, everything about it, starting from sort of the overall structure, the gross anatomy, going right up to sort of just a couple of slides at the end of just how it might be involved in motor learning or motor control. But there's obviously decades of work done in the cerebellum. If you go and speak to Yossi, you'll find out how many years of work we've done. As we were just discussing at the start, there's still as many open questions now as there was when we even started. So this is purely just going to be a little bit of a snapshot of all the different aspects of cerebellum, cerebellar processing, but it's not even going to touch the surface of how much information is out there, but it'll at least give you sort of an understanding of where to go and look if you're interested in different aspects. And then in the second lecture, I'm going to focus in on looking at information processing in a particular cell type and how that's regulated in vivo. So this is hopefully will just give you the background that we'll then move on to talk about the specifics later on. So we'll just cover, it really is just going to be a scan over the surface, but first of all, which is always important, there's lots of people in the field question how important the cerebellum actually is. We can function perfectly well without a cerebellum, so it's always the argument of why do we need a cerebellum in the first place. So we'll go over that. Then just quick gross anatomy, what the structure looks like, what the cell types are in there. It's a rather simple structure. Then on to go down into the specific pathways of where the information comes in. So mossy fiber to granule cell pathway, so that's the major input or the input layer to the cerebellum. Then go down into how this sort of first layer of processing is controlled by inhibition. And this is what we'll feature an awful lot more on in the second lecture I'm going to give. Then on to excitatory pathways, which come in and go up through the cerebellum, the parallel fiber, and all of the cerebellar pathway, which is what Yossi Yerom's interested in. Then how information is controlled by interneurons in the upper layers. And then finally, and very briefly, just touch on cerebellar-dependent motor learning. So, firstly, the most important question, I guess, why is the cerebellum important? So, to start to answer that question, we really have to go back to one of the sort of pioneers in the field, a gentleman called Sir Gordon Morgan Holmes, who was a field neurosurgeon in World War I. So he was sent out into the field just behind the front line. He would set up a field hospital, and he would get all the casualties that were actually coming back in. And he was documenting just the kind of deficits that some of these patients had. And they could have very specific lesions because obviously they were getting either shrapnel damage or shot in very particular places throughout the brain. So he could actually map where the damage was and what kind of symptoms or deficits that these patients were actually showing. And what he started to document, he became very interested in was patients who were coming in that had gunshot wounds or shrapnel damage, specifically to the back of the head where the cerebellum actually sits in a human. And he started to piece together all this information. He could see that the patients who were coming in, although their motor movement was disrupted, they could still actually move about. So they could locomote and they could move and interact with the 3D world. But what they found was that these patients would have very jerky movements. So they would be able to actually, so if they needed to walk across the wall, they would manage to do it. It would take them a long time, but the fine sculpted motor movements were lost. And they had these really jerky, uncontrolled movements. So he started to generate the idea that the cerebellum wasn't necessarily involved in generating motor commands and actually initiating the motor movements, but it was much more involved in the fine sculpting or the fine tuning of those motor movements. And that basically spawned decades and decades of work that we've all been interested in trying to understand what cell types are in there, how they actually process information, and really we've got an understanding, but we're still far away from actually understanding how it is that the cerebellum actually sculpts what would be the motor commands that would be coming from the motor cortex. And I think that's still a large open question in the field that we're all trying to still get at and trying to address with new techniques. So Gordon Holmes was the first one to really try and or really initiate that enthusiasm in people and trying to understand what the cerebellum was for. And in terms of what it means to both animals and patients, so both these examples, here's a cat that has a cerebellar ataxia. It's an inherited form of dysfunction or disruption of the cerebellum, where actually one of the main cell types of Purkinje cells lost. And you can see, as was documented by Gordon Holmes, the cat can still move. Its intended target, it'll just come into view in a minute, but here's its bowl of food. So it's a hungry cat, and it just wants to go across the room to get to its food and eat. But you can see its ability. It can still move, and it can still get to its intended target. But you can see how difficult it is for the cat to actually get there. Its movements, it can still move, but the actual gait and the way that it actually moves is severely disrupted and severely abnormal. And it's not just in animals, so exactly the same as another form of ataxia. Here's a patient here that's been held up by the physician. He's holding onto his arm, and he's just been asked to put one foot in front of the other, as would be a very simple task for us to do. But if it wasn't for the physician him actually holding him and holding him upright, he would fall over each time. Because his posture, his balance, and his ability to do those fine coordinated one foot in front of the other motion is disrupted. So in terms of from the animal point of view and definitely from the human point of view, the personal and societal impact of having this kind of dysfunction of the cerebellum can be quite profound in what would be simple everyday tasks that we would perform. So cerebellar ataxia, or disruption of cerebellar function, can occur or arise from a variety of different routes. Gordon Holmes was talking about the focal lesions, but this can also occur in car crashes or any other type of traumatic brain injury where you get damage to the back of the head or the back of the skull. You've probably all seen it, but in terms of alcohol or substance abuse, if you've seen somebody who has chronic alcoholism, you can usually see that they've got an odd tremor. And a lot of that is because there can be degeneration and disruption to the function of the cerebellum. You can get non-hereditary cerebellar ataxias, which is usually from substances of abuse, or again, if there's traumatic brain injury where you get cerebral edema or swelling of the brain because there's an increase in the amount of fluid that's in the brain. And then you get the hereditary types, which is passed down through generations, such as spinocerebellar or Fragile X-associated ataxia. So that's the sort of different forms, and that's I guess why the cerebellum is important, because if you get disruption or you get dysfunction or damage to the cerebellum, it can have a significant impact on your ability to actually interact or navigate around your 3D world. So that's the defense we use in terms of trying to get grant funding and be able to say why it's important that we really try and understand right down to the signal processing, because there's so much unknown. We were talking about this just before the lecture, that it seems that every decade that goes on, there's just more questions. The cerebellum, and we'll go on to that in a second, it's such a simple structure, but there's more and more questions because we really just don't understand what the cerebellum is actually processing it and why it's processing it. So one of the main reasons why the cerebellum is a complex structure to understand how it works is because it's effectively an interface or a hub where information comes in. Because the cerebellum receives vestibular information, so in terms of tilt or in terms of rotation of the head or movement or where the body actually is. It gets input from the amygdala and other places. So in terms of your emotions, it processes information, depends on what their environment and where you are in an environment actually means in terms of motor control, how you want to interact with your environment. It receives direct auditory input, receives visual input. It gets the descending motor command that comes from the motor cortex to say, okay, that's the intended movement. It compares that motor command with sensory information that's coming back from the periphery. So things like joint angles, proprioceptive feedback. And it basically processes all that different information to then send out commands that will fine sculpt motor movements. And I'd always just show this movie because both the motor cortex and the cerebellum are linked to produce or be able to produce what are amazingly complex movements in the human. And that's just it's an amazing repertoire of skills that we can actually use to coordinate our muscle movements to actually navigate our 3D world and interact with our 3D world. So the cerebellum is difficult in terms of understanding what it's processing because it gets all this amount of information. So how do you start to actually strip that apart and piece together what is the important bits of information and how does the cerebellum actually process that information? And in the second lecture, we'll look at a specific example where we're looking at somatosensory and the processing of somatosensory information in the input layer of the cerebellum. So, in terms of the gross anatomy, I said it was a relatively simple structure, but in terms of the sort of overall structure, this is a mouse cerebellum here, and it's split into different sections where the central section here, right down the sagittal plane or right down the midline, is classed as the vermis, and it gets a lot of somatosensory information. It gets visual input. It also gets auditory input. Then you have the paravermis heading out towards this section here, and then out into the hemispheres with crus 1 and crus 2, and also the paraflocculus. So the paraflocculus gets vestibular information, and crus 1, crus 2 gets a lot of somatosensory information from the periphery. And the examples I'm going to show, or the talk I'm going to give on Thursday, is about somatosensory information into the cerebellar hemisphere crus 2. And if you take a sagittal section right through the center of vermis, right down midline here, you can see the sort of structure here where there's a lobular structure where the cerebellum has 10 different lobules, starting off right down here, where this is the superior colliculus in the back here, and this is the brainstem. So you can see lobules 1 running through right down to lobule 10. And previously, maybe going back a decade or two, when people were doing slice electrophysiology, you would basically, and I've done this before, that you use this slice as one unit. So you go and record from one of the cell types in lobule 5, and then you go to lobule 9, just wherever the cells are nice and healthy, and you'd make conclusions about how Purkinje cells, or how a cell type processes information, or how it's what its electrical excitability is, and you just lump all those cells together, and you publish a paper on this is how the excitability of that cell is regulated. But as time goes on and we start to really, with molecular genetic mapping techniques, we start to understand where information is coming in, we actually start to see that each of these lobules are completely different processing units getting completely different information. So to lump all these cells together and say this is sort of how that particular cell type actually functions is a little crude. But one of the limitations we have, even with advanced molecular mapping techniques where we can really go in and map all the inputs that are coming into the cerebellum, only now are we really starting to get an understanding of where the neural map is, or what it looks like, and in terms of segregating what information comes into lobule 10 versus what information comes into lobule 3 or lobule 4. So that's still a big outstanding question in this field, is really mapping where all the connections are and what that means for the function of the cerebellum. So if we strip it back to the actual cell types which are present in the cerebellar cortex, why people thought the cerebellum would be interesting to look at is effectively there's only really 5 main cell types. The main output neuron, which is classed as, let's say, the integrator, which receives all the information that's coming into the cerebellum, is the cerebellar Purkinje cell. So a large soma with a huge, extensive dendritic tree, which gets all excitatory and inhibitory information, and integrates this to then choose the output spike pattern that actually leaves the cerebellar cortex. So it's a sole output from the cerebellum. So input or information comes in via 2 different excitatory pathways. The first is the mossy fiber pathway, and this brings information from the brainstem. It's usually sensory information coming from the brainstem, from the pons. These are glutamatergic fibers, so they're releasing glutamate onto the first cell type in the input layer, which is called the granule cell. And the granule cell layer, which is classed as the input layer, contains, well, in a human, on the order of 1 billion granule cells. So it's by far the most abundant cell type in the brain. And to a certain extent, that's why it makes understanding how information is processed in the cerebellum pretty difficult, because there's 1 billion granule cells which are each sampling different information coming from, different afferents are coming in. So that's where the major push is at the moment, to really try and understand if there's a structure to the granule cell layer. You've got 1 billion granule cells. Now, how do you organize the information coming in there that's actually going to be meaningful in terms of decoding at the Purkinje cell layer? So mossy fibers bring excitatory information in. These granule cells are small. They're only 5 to 8 microns across, and they have really, on average, 3 to 4 small stubby dendrites. And all the information that comes in, the synaptic information, comes in on the end of these stubby dendrites, so they call them end feet or claws. So the excitatory information comes in, drives excitation in the granule cell, and then there's an ascending axon which rises up through the granule cell layer, through the molecular layer, and then bifurcates to produce a parallel fiber. And this parallel fiber runs the length of the cerebellar cortex and can be on the order of about 2 millimeters in length. These parallel fibers then synapse with the more distal dendrites of the Purkinje cell, providing excitatory drive to the Purkinje cell, which can then increase spike output, which then leaves the cerebellar cortex. But in terms of processing information, it's estimated that a single Purkinje cell receives on the order of around 200,000 inputs from different parallel fibers. So each parallel fiber forms a single synapse as it goes through. So the dendritic tree is there, the parallel fiber courses through the dendritic tree, and on average just makes a single synapse as it passes through. But then the parallel fiber is actually going for 2 millimeters, so it can pass through many different Purkinje cells as the Purkinje cells are stacked along the sagittal plane. So that's one form of excitation coming through mossy fibers, sensory information. Then the other other pathway where excitation comes in is through the climbing fiber pathway, which we'll discuss in a little more detail later on in the talk. But the climbing fiber, each adult Purkinje cell receives input from a single climbing fiber. But where the mossy fiber or parallel fiber pathway, there's 200,000 inputs, this single climbing fiber actually wraps around the more proximal dendrites of the Purkinje cell and makes many, many synaptic contacts. So it's a really strong, profound synaptic input that comes from the climbing fiber, which originates from the inferior olive. So these are axons of inferior olivary cells. So that's the two forms of excitation, mossy fibers and climbing fibers. In the granule cell layer, the sort of excitability of these granule cells, obviously it can't just be driven by excitation or you get runaway excitation. So this is controlled by granule cell layer interneurons classed as Golgi cells, which are GABAergic and provide the inhibitory input to granule cells. So this sort of controls the excitation coming from the mossy fibers and regulates the excitability of the granule cells. And I'll go into a little bit more detail in the second lecture about exactly this, the sort of balancing between the excitation and the inhibition coming onto granule cells. Also, in the molecular layer, where the dendrites of the Purkinje cell are, you have another two types of interneurons, stellate cells, which are on the sort of middle to outer third of the molecular layer, and basket cells, so-called because they form these baskets around the soma of Purkinje cells. These two molecular layer interneurons then also are GABAergic and provide the inhibitory input to the Purkinje cell. So that regulates the excitability and sort of dampens the excitability that would be coming through the parallel fibers in the climbing fiber. So that's the 5 main cell types that you'll see throughout the entire of the cerebellar cortex. So, yeah.
Is there any correlation between the size of the cortex or the analogy of the cortex in non-mammals and the size of the cerebellum?
Whoa. There's a profound question.
You can take growth inhibitors.
Yeah.
It's
No.
like 1-to-1 ratio in terms of, I mean, it's not 1-to-1, but it's like there's a linear curve with slope of 1, while the cortex grows, the cerebellum grows proportionally. That's what I know.
I mean, in terms of size-wise, yes, but in terms of functional growth, you know, why they're growing together and they are the same size, I mean, we really haven't, I mean, how many years have we been going after the cerebellum, and that's what we'll go into here. But only now, people like Tom Jessel have got these nice molecular genetic techniques to really start to go, okay, let's look at this input pathway and see where it goes. Only now are people starting to go after the efferents copy that comes from the motor cortex, because obviously, if there's a motor command going out, the cerebellum needs to know. It's a bit more complex than we're going here, but there's internal and external models where there's an internal model that says, okay, the motor cortex said that was the intended movement. The cerebellum gets that. This is the hypothesis. The cerebellum gets that information, and then it gets feedback, sensory feedback from the periphery, which says, okay, you intended to put your left leg out by whatever distance, but in actual fact, you just bumped into something which pushed it out of the way. So the sensory feedback, there's a mismatch between the intended motor command and the actual sensory feedback, and that's what the role of the cerebellum is thought to be about, is about being able to correct that, to update and fine sculpt that movement. So even now, people are going after the efferents copy. We still don't know what efferents copy, what information has actually been sent to the cerebellum. So the cerebellum for years is, it's all about motor control. We haven't even mapped what information it's getting from the motor system. So it's this sort of disparity where people go, yeah, who cares about the cerebellum? It's stuck there at the back and we don't really know what's going on because we really don't know what information it's actually receiving. So in terms of, size-wise, they grow together. But in terms of functional, what's the relevance of having the same size cortex as the same size cerebellum? So that's still open, still unknown. But naively, going back in the day, a few decades after Gordon Holmes identified that the cerebellum is important for movement, people said, okay, there's only 5 cell types in the cerebellum, and so this diagram shows that in the sagittal plane, it's just repeating units of exactly the same. So you've got Purkinje cells here, you've got your inputs coming in, and then as you go along the cerebellum, it's exactly the same. So I said, okay, 5 cells, repeating units, anywhere in the cerebellum, whether it's the vermis in the middle or the hemispheres at the side, it's exactly the same. So in terms of understanding it computationally, it must be pretty simple. That was the initial thought. But as each decade goes on, we seem to ask more questions than we actually answer in terms of knowing specifically what the function is. And the reason for that is if you actually start to go down, and as I sort of alluded to before, it's a sort of hub of information. And even just looking at the somatosensory input, and this is just in one brain area, so this is in CRUS 2, which I'll talk about on Thursday a little bit more. If we even look in this one area, just looking at one type of sensory input, or at least somatosensory input, if you look at where the inputs from stimulating different areas just on the face of, this is a rat, whether it's the whiskers or whether it's on the nose or the ears, there's a fractured somatotopic map that depending on where you're recording from, there'll be strong input from that particular area. But in terms of trying to work out how there's, whether there's a specific topology in terms of understanding where that information is set out, it's just not that easy. And that's just for somatosensation. So you've also got visual input, auditory input, vestibular input, so on and so forth. So it can be, even at this level, can be an entirely complex map of where all these inputs are coming into. And really, we don't have a really good, detailed, defined map, similar to what you get, let's say, in the visual cortex or in the auditory cortex. We just don't have that for the cerebellum as yet. That's for input that would be coming in through the mossy fiber pathway. Then it gets a little more complex as well in terms of that the cerebellum is split into what are classed as microzones. And the other excitatory pathway I talked about, the single climbing fiber that comes in. So each of these microzones are innervated by different climbing fibers. And they seem to project to specific microzones for presumably specific computational reasons. But the cerebellum is split into these microzones, and each of these microzones receives very, very different mossy fiber and climbing fiber input. And even more complex is the fact that each of these microzones then converge down on the output. They converge to different centers in the brainstem. So we don't know, it's complex at the level of we really don't know where the information is actually coming in. Let's think about even at the granule cell layer, billion granule cells. How do we actually map function to function, as in the movement of the animal to actual what information is coming in, is how that's actually being processed? Did you have a question? So there's still big debate about what information is the main sort of pet theory is that it's bringing information about error signals. So I'll go on at the end to talk about that. Actually, climbing fiber is thought to be a sort of instructive or teaching signal. So the mossy fiber is bringing lots of information about where you are in space and 3D space and so on. But then, let's say if you've got an intended motor command that you're going out and going to pass something, and you step over something or something is in your way, there's a mismatch between your intended motor command and the sensory feedback. And there's a lot of evidence out there that the climbing fiber actually sends the signal to instruct the fact that that was an error, and it should take specific notice of that. And the climbing fiber is very also pivotal to a variety of forms of plasticity, because it's such a, it's a single climbing fiber comes in, it's a really strong input that actually, if it's a teaching or instructive signal, the climbing fiber is the sort of the gate between plasticity induction and no plasticity induction. So in terms of cerebellum dependent motor learning, it can be really quite important. And there's one example I'll give at the end about Pavlovian conditioning, that you'll see the pathway where the climbing fiber is actually useful in terms of providing a plasticity inducing signal. But again, still in debate exactly what its role is. So there's lots of levels of complexity, which is why you can get effectively such a simple system with a limited number of cell types, but the level of complexity comes with the sheer enormity and divergence of information that structure is actually receiving and has to process in some form of meaningful way. So let's go down into the actual pathways. And look in a little bit more detail, give you some details of that. So we've talked about the mossy fiber granule cell pathway, which is bringing that sensory information up from the brainstem. And this is the pathway I've shown here, which is important in terms of propagating information down to the Purkinje cell. But I think this is even more of an example, or it's a good example, of that level of complexity. So this is from the Brainbow mouse from Josh Sains in Jeff Lichten's lab. And I put this up here just because I've shown the different levels of complexity in terms of the somatotopy and the microzone orientation. But even if we just go down to, so this is a snapshot, a confocal snapshot from the granule cell layer. So that's the input layer of the cerebellum. And with their Brainbow mouse, each of the mossy fibers that are coming in are colored in a different color. And this is only 100 microns by, I can't remember, it's about 20 or 30 microns that way. 100 microns by 100 microns. And you can see the number of different mossy fibers that are coming in. Each of those mossy fibers will be conveying a different set of information. And all that has to be processed by the granule cells that are present in here to then go up and converge on the Purkinje cell. All that information will converge, and then that will regulate the output spike pattern. So even if you look at the gross anatomy, right down to the actual anatomy at the single fiber level, you can see the complexity even at the input layer. So this brings somatosensory information or other types of sensory information into the granule cell. And the mossy fiber itself is quite a unique structure in a way, that mossy fibers are about 5 to 8 microns across. So they're a single axon which comes to a really large head. And a diagram taken from Eccles' work, in grey you can see the mossy fiber terminal here. And in sort of pink red are the dendrites of the granule cells. So a single mossy fiber can synapse with, on average, about 20 to 50 different granule cells. So when you get activation of a single mossy fiber, you almost get a cluster activation of a number of granule cells within the granule cell layer. Yes. As in the mossy fiber has. Yeah, so the mossy fiber, it's not just a single mossy fiber that comes in, so they bifurcate and they go to different areas of the cerebellum as well. But again, that just makes it, even anatomists haven't mapped that yet. So in terms of us functional neurophysiologists trying to work out, okay, so you look at let's say a slice of the cerebellum. Where are those mossy fibers coming to, and what are those functional modules? Why why is there 20 or 50 granule cells activated here and across there? How does that information then converge at the next level? That's sort of the sort of real push in the field, is to really understand that structure.
Why do you have the claw?
The claw on the granule cell? So again, it's probably because it's the unique structure of this. So there's the mossy fibers there, and the granule cell dendrite comes out and has a claw because it makes more than one synaptic connection with the mossy fiber. So you can see all these little dimples that are in here, and you can see, well, there's one granule cell dendrite, and it's sort of making that. So it's trying to make that a really strong connection. But it gets a little complex. But this single mossy fiber doesn't necessarily have exactly the same release probability of each of those active zones. So there can be heterogeneity in, it's not just a case of this fires and then there's an explosion of activity. There's actually a sub-regulation here as well, which I guess increases the computational importance or significance of that. When you have a relatively simple granule cell that only has 4 dendrites, where you can have other cells which have got enormous dendrites, then the complexity comes with the way that it's set up anatomically. So the granule cell dendrites are pink, and then you have, in yellow, you have the Golgi cell axons which come in, which provide the inhibition. So the excitation is coming from the mossy fiber in grey, and the inhibition is coming from the Golgi cells. But also the mossy fiber terminal itself has another sort of uniqueness to it, that actually the whole thing is ensheathed in a glial sheath, which is quite interesting in terms of once you then get transmitter release within from the mossy fiber, then it's actually trapped within this glial sheath. So you get some interesting spillover or spill out effects of the neurotransmitter, which can produce different inputs to the downstream granule cells. And there's only here, and I think in the thalamus is the only other place that you get these sort of rosettes. So there's obviously anatomically a really good functional reason for this, but there's only 2 real areas in the brain that show these glomeruli type structures. So, yeah.
A mossy fiber can connect 20 to 50 granule cells. How many mossy fibers does one granule cell connect to?
4.
4.
So the granule cell has 4 dendrites. Well, on average, 3 to 4. Let's say you can get ones that have just got 1 or 2, and then up to 5. So on average, yeah. And again, as time goes on, the debate swings one way and the debate swings the other way. So Henrik Jorntel in Sweden did some experiments in decerebrate cats where he was mapping receptive fields, and effectively what he said is that one granule cell with its 4 dendrites basically get exactly the same type of information coming into that one granule cell. So basically that one granule cell is, let's say, in a simplistic sense, it's a proprioceptive granule cell. The other one's a visual granule cell. The other one's an auditory granule cell, which went on for about 4 or 5 years. And then Adam Hantman, who works in Genelia Farm, and used to work in Tom Jessel's lab, so he's been developing these really good and unique molecular genetic mapping tools where you can go in and use virus and then really map specific input pathways. And he had a paper in eLife just recently in the last year, which categorically said that a single granule cell can get input from the pons, it can get input from vestibular, it can get input from here. So they're integrating, even one granule cell is integrating information that's coming from different sensory modalities or different modalities. So even down to the level of a single granule cell, which is a billion of, that granule cell is computing or integrating different forms of information to choose whether it actually spikes or doesn't spike. As it seems to go on, as time goes on, people debate it one way and then it comes back the other way. But it definitely seems to be that that particular areas have a preference to process a majority of one type of sensory information, but it's definitely comparing that with other forms of sensory input. Okay, so in terms of being able to record what this looks like, and it's what I've been doing both when I was in Mike Heuser's lab, when I was working with Mickey in London, and then what we've gone on to do in my own lab is doing in vivo pants clamp recordings. And in terms of just showing what type of inputs you can actually get, this is a recording from an anesthetized rat. We're recording from the cerebellar hemisphere in CRUS2, and we're giving ear puff stimulation to the whiskers and the upper lip, just to give sort of somatosensory input, which gives a strong input to CRUS2. And what we managed to do at that time, using blind patch recording techniques, is we actually managed to record from the presynaptic mossy fiber terminals. So actually recording directly at the release from the mossy fiber and what kind of pattern of activity. And in blue, you can see where is the sensory stimulus or the ear puff that we gave. And when we give that brief 60 millisecond ear puff to the whiskers, you can see this burst of action potentials which are invading the presynaptic mossy fiber terminal. And then we could record from, not simultaneously, but we could record from the postsynaptic granule cells to then see what type of or the pattern of input that this sensory stimulus would actually give. So here's a mossy fiber, and then when we're recording this was in current clamp, and when we're recording in voltage clamp, the same sensory stimulus, you can see this burst of excitatory postsynaptic currents, which is effectively mirroring almost one-to-one the activity that you see in the presynaptic terminal. So it allows us a way to actually look at the presynaptic function and how that is converted into transmission to the postsynaptic cell. Yeah. Is there?
Presumably in the granule cell, after the blue,
Yeah.
is that presumably from other mossy fiber inputs?
So we believe that it's only coming with our stimulus. We're only really giving it one form of information. So from all the experiments that we're doing, we kind of concluded that it was just coming through a single mossy fiber. So this is a presynaptic recording, so it can only be
Thinking of the other one.
here.
The
Yeah, I mean, that's one example. If you take it across and look at the PSTH, then there isn't necessarily a double bump. In terms of if that's reverberative activity, it might occur, but it's not consistent to say that it's multiple mossy fibers coming in. So we think that our stimulus is just activating a single type of mossy fiber. That input is coming in, and that's what we record postsynaptically. That's in CRUS2, so that's where there's a strong somatosensory input, but some rather beautiful work done by Alex Arendz and Troy Marguerite in London. They did quite a cool thing. They, in anesthetized rats, And what they were interested in was the vestibular input to the flocculus. So they're recording under anesthesia, recording from granule cells. And what they did was that, obviously, vestibular input, you need to tilt, rotate, or move the head to trigger that vestibular input from the movement of the fluid in the semicircular canals. But what they did was that they actually mounted the rat on a movable plate. So then what they could do is they could patch onto a granule cell and then actually start to rotate, while still patched and recording whole cell, they could rotate the entire animal to then activate in a particular axis the vestibular input to that granule cell. And what they found was here's the sort of movement of the rotation of the plate and recording from a single granule cell, that actually the granule cell would have a preferred direction. So this is under baseline, then they would start to move the plate, and actually the input through the mossy fibers to this granule cell would shut off, and then you would come back through a different angle, and then you get a really strong input when it was in a preferred direction, then it would switch off again, and then back through to switch back on again in terms of input. So it's almost as granule cells in the flocculus have a preferred direction that they're processing information when the animal is in a particular head orientation. What they didn't do in this, which is a bit of a shame, is because the frequency of this input is really quite low. So what they didn't look at is what the spike output, which would have been really nice to have a look at. Okay, how this is the input that the cell is getting, but obviously it's integrating that information. What does that mean in terms of the spike output pattern? And what I guess you would expect is that you have a peak in the spiking here, goes down to silence. They get another peak in the spiking here, which is higher, comes back down to silence, and so on and so forth.
Can I ask a question?
Yeah.
First, did they mount the whole recording apparatus onto the moving platform, or how is it technically possible to do that?
Yeah, basically the whole system moved. So the recording electrode, the amplifier, and everything on top was just moving as one.
And are these cells change detectors, or so if they move a little bit and then stop, do they pertain firing or do they cease?
Oh, that they didn't know. That they didn't do. I'm not sure, because this was the first time that actually anybody had done that. Don't know if anybody knows that. You mean whether they would just maintain their firing at a constant rate if they stopped midway between here?
Right, so if they are signaling that there is a certain angle, or if they are signaling that we just arrived at it.
Interesting question. It's not something they did. I know how painful these experiments were. So this was a nice science paper at the time as proof of concept and to get some really nice, meaningful information. But moving your manipulator and your recording electrode at the same time as the animal, or just moving a glass of...
Once you manage to do this difficult technique, probably it's not that difficult to find out whether they react to the change of the angle or to the angle itself.
Yeah, it depends what yield is like.
Yes.
So if yield is low, yes, but definitely you can. I mean, that's exactly the kind of things I say to postdocs in the lab. Yeah, I know the yield is extremely low, but if you keep going for long enough, you'll get your data. At some point, you have to make a decision to go, okay, the reward doesn't pay for the amount of time you've got to invest to do it. So they have been working a little bit on this, and there's so many different ways you could go on this. And what they did actually start to do, I think, is do this way as well. So they spent the time, they've done that part, they wanted to do this, and then I think the idea was once you've got all that, but there are some severe technical difficulties with doing all of those. So I'm not sure to what extent they're continuing with this. But it's a great question. Great question. Okay, and
I can comment that there's a related question that there's a guy in Yossi's lab. I don't know if you know, a young, tall guy with a beard.
Yes.
So he's working on not on the cerebellum but on the DCN in the auditory system, which is a cerebellar-like system, actually similar in some ways to the cerebellum. And they are doing something similar in terms of like moving the rat or moving the auditory signal.
Okay.
So and comparing the two. So it's actually related. So you can go and talk to him about things like that.
And that's a mounted moving plate with everything mounted on top.
It's not whole cell. It's a cellar recording, cellar touch kind of. I mean juxta.
Yeah.
Because it's about I don't know 5 millimeters deep.
Yeah.
So it's
Was that deep cochlear nucleus?
Yeah.
Okay. Yeah.
Very deep, so it's difficult.
I mean, that's tough enough. But I mean, in terms of spike I mean, this is really, it's really interesting, but in terms of understanding cerebellar processing, to a certain extent, you don't want to know what the granule cell gets, because if it doesn't spike, the information stops there. So actually the important part is knowing what the spike output to that is, and then you start to understand how vestibular information is actually, but that's first pass. It was heroic work, so it's definitely good from that point of view.
So I was just wondering, so the rat,
You want it back?
will it orient itself using the environment around it?
I can't remember if it was.
What do you mean?
I think it might
It's
have been.
inspired when it's here. Is it because the environment is a certain way when looked at from here?
So I mean, my interpretation here is that it's probably not like it's an anesthetized mouse, a rat.
Yeah.
So it doesn't really know what it is in space. So that's why I would bet that this is something to do with the movement, rather than, I mean, actually you get like vestibular input during the movement rather than being fixated on a certain position, where basically you're unconscious, so you don't really understand what's going on.
Because they probably did it in dim light anyway, but in terms of visual input or whatever it's getting during that, you really have to, in an anesthetized mouse or rat, you really have to shine LEDs in its light in its eyes before you get a visual input in the cerebellum. So it's there, but it's a strong stimulus you need to give before you actually see it.
Vestibular is much stronger than?
I would imagine much, much stronger, yeah. Especially because they've gone to the flocculus, which has been known for a long time to really prefer. So I mean, if you go to the vermis or you go to Crus, the likelihood of seeing any vestibular input is almost zero, because it seems to come in. That's a specialized structure which seems to have been developed in the cerebellum for dealing with this, or the majority of the vestibular information that's coming in. Okay, so just quickly, as I say, we're going to focus much more on the second lecture on the sort of transformations that occur in granule cells. But just to say what we can do with in vivo patch, in granule cells are great because they're only 5 to 8 microns across, so they're electrically compact. They're isopotential across all this, so you can see it basically as just one big ball. So in terms of being able to control in voltage clamp, you can get really nice clamp of the cell. So you can record, in both voltage clamp, you can record the EPSC input you're coming, and then in the same cell, switch from voltage clamp to current clamp to see what the same sensory stimulus then produces in terms of overall spike output. And the bit that I'm going to sort of focus on a little bit more is, we talked about Golgi cells are the sort of the brake in terms of excitation to granule cells. They provide the GABAergic inhibition to temper the mossy fiber excitation. But what we can do is we can use pharmacology, which technically is not a simple or trivial thing to do. But you can use pharmacology in vivo to knock out particular receptor types, and then have a look at, let's say in this, we use gabazine here to block GABAergic inhibition, and then you can look in control, apply your drug, and then look at the effect of blocking a particular receptor subtype on the excitability or the input to output transformation that these cells can actually produce. In terms of if you are thinking, or you do go on to do patching or in vivo experiments where you want to do pharmacology, then just a word of caution that it's extremely difficult. The problem is that you just don't get washout. Once you put the drug on, it just starts diffusing across the brain, and you can't really stop it. So in terms of yield, cost, benefit, my original degree is in pharmacology, so I'm a pharmacologist by trade, but I have now, to all intents and purposes, stopped using pharmacology. So we're now using molecular genetic techniques. So the data set I'm going to talk about, that myself and Mickey were working on, for one drug, took approximately 6 to 8 months to get an N of 6. And that's recording 4 days a week. But the problem is, any way that you apply a drug, you change the bath volume that's on the top. And once you've got your cell, if there's any mechanical movement of the recording patch, you get a change in your recording integrity, and you've already put the drug on. So if you get a change in recording integrity, you have to terminate that experiment and go and start another one. And about 60-70% of the times, the recording configuration would change roughly about 2 minutes before you're about to take your second recording. So just a word of caution. It's definitely doable, and in different brain areas it's definitely doable, but there are molecular genetic techniques that are out there that might save yourself an inordinate amount of time. Okay, so that's what we can do. As I say, I'll go on Thursday to talk in a little bit more detail. So that was the mossy fiber input. And just to sort of give an overview in terms of are Golgi cells important? Why are they important? They're interesting. I'll give the sort of quick introduction now, and we'll go over it a little bit more on Thursday. But granule cells are kind of interesting. Oh yeah, get some coffee. Yeah, let's go. We're halfway through. It's good.
Can you maybe also the slide we discussed again? Hier, der das ist die Geschwindigkeit, glaube ich. Und dazu passt es ja irgendwie am besten die nicht, genau, die ist nicht. Aber das ist Geschwindigkeit und das passt irgendwie genau zusammen.
Also du würdest sagen, dass hier in der Mitte eine höhere Feuerrate ist als am Rand der Objektivsäule.
Und hier hast du noch eine Projektile-Kurve und das ist schmaler.
Das heißt, da steht. Ja.
Das ist meine Intuition und deswegen habe ich gerne mit meinen Sachen nicht drauf, weil es halt nicht
Genau,
optisch
das
sehr
hat
anders
mit der
ist.
Position passiert, ja. Genau, ja. Ja. Ja, oder es ist eigentlich, kann man auch sagen, einfach Geschwindigkeitsspektrum, oder?
Aber Geschwindigkeitsumrichtung, weil ich meine, wenn es da ist, wenn es
Hier ist die
hier ist das
Geschwindigkeit genauso groß, aber mit einer anderen Sportzeit.
genau.
Naja, oder? Das stimmt aber auch, dass die Feuerrate hier, wo die grüne Linie die Nullschneide durchschneidet und hier auch wieder, wo die grüne Linie die Nullschneide.
Aber ich meine, da ist ja die Bewegung ist ja dieselbe Richtung. Also das ergibt ja irgendwie Sinn, dass hier
Naja,
quasi
aber
dieselbe.
das Experiment gibt nicht genug Daten sozusagen, weil die Geschwindigkeit und die Position jeweils gleich sind, hier und hier. Also hier die Geschwindigkeit ist hier negativ maximal und die Position ist null und die Geschwindigkeit ist hier negativ maximal, aber die Position ist auch null. Man hätte nochmal diese Geschwindigkeit haben müssen, aber an einer anderen
An einer anderen
Position,
Stelle.
um das zu sehen.
Aber wenn du die große Zeit nimmst, ist es schwierig. Achso,
Ja,
du
man
meinst eine
unterschiedliche Beschleunigung
andere
nochmal.
Zeit.
Ja, oder irgendwie, oder einfach mal hier anhalten und hier die Geschwindigkeit auf null setzen.
Man muss doch nochmal anhalten.
Ja, genau, ne?
Wenn man schon so
Ja, das sind ja nur 7 Sekunden, also sieht jetzt ja nicht so aus, als wäre es so schwer gewesen, dann nochmal 7 Sekunden anzuhalten.
Timeskip Study.
Ja.
Um zu wissen, wie schlimm das wirklich gewesen ist. Ja, weil
Ich meine, jeder einzelne Teil ist wahrscheinlich wieder extrem viel Arbeit, weil man wieder alles neu aufbauen muss, aber den einzelnen Fall nochmal länger zu
länger zu
machen,
machen.
ist nicht so schlimm. Ne? Oder wenn es nur 7 Sekunden lang sein darf, hätte man ja einfach mal die doppelte, also den gleichen Zyklus zweimal in die Sekunden stecken können. Und wenn man nur 15 Trials von diesem Experiment gehabt hätte, wäre es ja auch genauso gut gewesen. Die Daten sind ja so sauber, dass es auch trotzdem schön gewesen wäre, wenn es nur allzu viel. Naja.
Naja.
Vielleicht ist es jetzt auch nur, ja.
Really?
They think this graph cannot
Waiting for
exist.
Jackson for the rest of the game.
You cannot plot a function that would have this face of non-dimensional
I
spikes.
know you're trying.
And they were kind of
So this
saying,
is,
this is that
I know we
phase
need
of
an
the
extra
of
worker
the
for that
spike,
too.
and this is that phase.
You gotta know
That's
that was
not
kind of nice. That's
was
why
really
not
hilarious.
perfectly consistent. Okay.
So what, if
Mine
look at the
is
spike
just like one of
graph,
those things
look
that
at the
I'm like,
spike when it plateaus, so it had a
So they
VDP value
had
of
something like that. It's really,
above
really
zero.
kind of funny. So they would have something like that. It goes like, you have, this is the VDT,
Yeah.
and they had something like the voltage goes straight, and then goes back, and this is zero. So what the minimum voltage is, the derivative is still negative. So it should continue to go down, but it actually instead it goes back up, but it goes back up, right? And it actually crosses zero at an intermediate point, which
When
of
it's
this
not
phase.
peaking. when it's clearly changing
Basically,
voltage.
there's no function that can actually generate
Where
such
was that paper?
science.
Good.
That's what I'm saying,
Good. Weird.
Nice.
Weird.
But you look at this thing and you don't pay attention to you see that graph, and then you
I
see
spent
that
the
well,
entire
then
day
the paper.
reading that graph and analyzing everything that was
Actually,
in that
what
graph
I'm
before I
referring to
felt
there, can
instantly
you
young
imagine?
again. But I started
Yeah.
walking tiny
Then I went
and
and
yeah, and
told
so
one
and
of the
like
physicists
I
about
started
just
like
to make
writing
sure that
stuff
I
down
got
as
the
we
graph,
talked, and I was like,
It
this
was
is not
they,
funny, but
they
that's just what
published
I
it,
read it. So like yeah, like I googled it
Yeah,
on his blog
crazy.
and I sent him an email actually. I was like,
Like,
yeah, because I was
it
like,
comes down to
I
zeros
wanted
in
him
the
to
middle.
know like
Yeah.
that.
So
Yeah,
you
I
know
was
where
like,
the
hey, I
well,
asked for
fits.
you trying to
Yeah,
see what
that's
it's
the
value.
logic. Yeah. Whatever they're saying is
He
correct.
knows.
That's
He knows.
yeah.
That's what
That's
I
just
thought. Jesus, that's kind of
it's always just,
that's what I was
yeah.
wondering. I'm like, do
I'm
you know that
horrified.
you like are
I
entire
mean,
classes for
all
midterm
of
on
this
me?
part of the paper.
Well, what's the
Yesterday I was kind of also.
Yeah, right.
No,
Like,
it's nothing
I'm like,
loaded.
that's kind of creepy because I was like analyzing like
Yesterday I was
the like
talking
he
to
had this problem where like he really felt like bad
Martin.
about his age, and he was only
So
like
he was doing
26.
patching in a
He had
private
entered the
company
military
at
when he was like 18 or
asking
something,
him to
and he kept
do a
like
bit of a
mentioning this like
psych
old blah blah blah
modelization,
thing
psychic
where
acting,
he kind of like
things
psychoanalyzed
like
him.
that.
Like he was like, he was telling me he's really having like issues
It's not
like
a sensory
tracking all
neuron.
this stuff and like how this is
They're not
causing him to like
modulated. No, and
I don't
modulation
know,
but
you know,
I
like
even go
I feel like I
prefrontal cortex,
do.
motor cortex, they're all modulated.
Close? No, I don't think about
It's
you.
a function.
Like you're
Really?
So
like
so
you
now
get
what?
the instructions to
I didn't even
out
say
and
to
guys.
talking to Christina
I
to
did
do
you
normal
just
things.
really not
I mean, while we
Because
sit
visual
down,
is auditory, isn't it?
no, we sit
something
down
like guys.
and do things.
Yeah.
Very much like
But
that.
it's small, and I don't know,
That's cool.
maybe
Yeah.
we should just
You change that to what you think is
go and boil
a
meat to
really good research
another,
outcome. She gets something
step
coming from the school because the student is arguing with
somewhere
it.
else. Prefrontal
You say, oh
cortex is
wait,
complicated because it's like
exactly.
you
That's
know
like
you can't apply
that's kind
anything.
of the fun, you know. There's the symbolism that you take to it. That's like I remember I was reading Metaphor 51
I suppose what's
and the main character kind of
the
shares
originating
very
No,
I'm
I
drawn
thought
to
it was like
teacher subjects.
so
So
fun,
my English teacher in high school would talk
that they
about
can
like
now
how
just a much
nuns have
bigger
like a
meaning.
name at
uh,
the beginning of each
well,
contact
no, you have to go
symbol,
so
like
to,
to indicate
to
something new.
be
That's why it's
they
Monday, blah
have
blah
to
blah.
what is it full
So
time
anyway, while they're talking, I'm kind of
working
like
years close to
listening
So
and
who's
watching.
first
Like
and
I go to the
Papa?
end of the author
Oh,
section
sorry.
and then
where he
U
talks
is
about like
coming
how he wrote
close
this
to
book and
second
yeah, like they were like
L
all
is
paid by
potential
the
G
typewriter.
and
I'm actually
Just
like
pointing to
really
that.
terrible and
yeah. No,
uncreative.
but
And
Like
you
I
told
usually
me
just
that
kind
she
of
has
like
like
I
she has
mean this thing has
actually
to
much
be my
better.
typewriter and
Now she's much
Montag
more
comes from
accurate.
the native
No, but
pencil that I was
completely. She's
holding.
now like back checking Pyro's
And I'm just like
doing
reading
absolutely
this and I'm like,
nothing.
alright, like this is
So literally, yeah.
So he already left.
No, he's I've kept him on until next summer because he's got other things to do. He's just
She
overseas
lent
and
me
needs
this
to do.
book with and then also had annotations.
But yeah, so now you know the it's going to be so funny because
We know why they
Theo's
use
calling
different
back.
variations of
Yeah,
yeah,
I
it's the
communicate.
way
I
they're
mean,
cut.
you
No
know,
pun
yeah.
made up, put
It's
in. So
responding.
funny.
The
I
upper guy
agree.
in the We good to go again? So this is what I'm going to talk about a lot more on Thursday in terms of the inhibition or how inhibition regulates granule cell activity. But just to put it into, give you a bit of background detail and to put into perspective where the field is at understanding what kind of inhibition granule cells actually get. So granule cells are kind of unique in the fact that, yeah, they're this relatively simplistic neuron, 8 microns across, 4 small stubby dendrites, so only gets input from 4 different mossy fibers. But even at the level of inhibition, and probably due to the glomerular structure, where we said this large mossy fiber terminal activates 20 to 50 granule cells and is covered in this glial sheath, granule cells actually receive 3 different forms of inhibition. So the first type is tonic inhibition, which you may have heard about. And this is a persistent standing conductance, which is there effectively all the time. And work done by Zule Nusser and Mark Farrant at UCL, what they did was EM immunogold labeling, where they used 2 different sizes of gold beads. So they used large gold beads, and it doesn't really matter about the membrane biophysics, but for the alpha-6 subunit. And granule cells, alpha-6 subunit of the GABA-A receptor. And to cut a long story short, receptors which contain the alpha-6 subunit have an extremely high affinity for GABA, but a relatively quick desensitization time. And this extremely high affinity for GABA and their positioning, because if you look at where these gold beads are, so here's the granule cell dendrite, here's the Golgi cell axon, so the GABA is released, crosses the synaptic cleft, and synapses with the granule cell dendrite. These high affinity receptors are actually located peri or extrasynaptically. They're not in the synaptic cleft. And with their extremely high affinity for GABA means that they can actually sense GABA levels down to nanomolar range, so in about 100 to 200 nanomolar. And that is about the extracellular concentration of GABA. So they can sense this low-level extracellular GABA, and they're activated by this sort of, let's say, ambient GABA, and which leaves them open. And if you look in voltage clamp here, well, we'll go in a second to talk about what these are, but if you block GABA-A receptors with a selective GABA-A receptor antagonist, you can see that the standing tonic conductance is blocked, and there's sort of a decrease in the noise, which is the flickering or opening of these extrasynaptic GABA-A receptors. So that's tonic inhibition, which is a persistent tonic conductance. And then these smaller gold beads, which are present actually in the synaptic cleft, are alpha-1, or staining for alpha-1 subunit, which these are receptors which contain the alpha-1 subunit, have a moderate affinity for GABA and a very, very fast desensitization rate. So they're perfectly suited to sense high levels or high concentrations of GABA being released from the presynaptic Golgi cell terminal. But once they're activated, you get fast onset, but fast inactivation or deactivation time. And that's fast point-to-point or phasic communication, or phasic inhibition. So we have tonic, persistent, slow, fast point-to-point phasic inhibition. And then there's this third type of inhibition, which is probably as a result of having this rather unique glomerular structure, where when GABA is released, so if you get high-frequency activation of a single terminal, or activation of a terminal that's not directly opposed to that particular granule cell you're recording from, you can get spill out of transmitter from the cleft, which will actually activate receptors which are just outside the synaptic cleft. But because there's this glomerular sheath, and once GABA is actually released, it can't really go anywhere, what you end up seeing is this sort of elongated, extended, slow, or spillover type of inhibition. As I say, that's just a basic introduction. I'll go in, the Thursday talk will be really going down into what this actually means for information processing the granular layer in vivo. But just to give you a sort of background that there's a complexity in terms of what type of inhibition granule cells actually receive. But at present, why we actually started this work, we were doing it with Mickey, and a lot of this was done in London, and a little bit of it when I was in Edinburgh, is that from all the in vitro work, and there's been shown in many other different systems as well, what has been sort of concluded, or the present idea in the field is that if you get fast point-to-point inhibition, what it does if you look at the input-output curve, so the frequency of mossy fiber input versus the firing rate of granule cell, and this is in vitro, what they found is if you start to give, using dynamic clamp and a few other different techniques, if you give fast inhibition, what it does is it just shifts the input-output curve to the right. Whereas if you get this slow inhibition, and this is just sort of an offset, shifts the input-output curves and produces an offset. Whereas if you put this slow spillover inhibition or the tonic inhibition, it can actually change the gain or the slope of the input-output curve. And that's where the sort of field is, but this is purely from slice experiments where you actually have control over the type of input it gets and a modicum of control over the frequency and the timing of the inhibitory input that comes into these cells. And I think on Thursday we'll show that although some of this is still correct, the features of what inhibition actually doing is really quite different when you look in vivo and using sensory stimulation and not just artificial stimulation where you're stimulating the slice or using conductance clamp injections of currents. So that's what's known from decades of slice work. And there was an indication from work done by Paul Chatterton and Mike Heuser's lab in London that even spontaneous activity, so this is in vivo, in anesthetized rats, and just looking at spontaneous activity in the presence and absence of inhibition. So when you put on gabazine, that's a selective GABA receptor antagonist where you block all types of inhibition. Here's in control where you can see in current clamps some EPSPs, and these are spontaneous, randomly occurring EPSPs. When you block inhibition in the granule cell, then you can see that the cell becomes much more electrically compact, much more excitable, and now you get an increase in the amplitude of the EPSPs, and in some situations you get actually spontaneous spiking. You can see that in the bottom, where's a control trace, where because of the resting membrane potential, you get very, very low spontaneous firing rates. But if you block inhibition, cell becomes much more electrically compact, more excitable, then you start to get much more spontaneous spikes. So inhibition is controlling the level of spontaneous activity that's occurring in granule cells. And on Thursday we'll have a look much more at the evoked activity that we can see in these cells. And this is basically where the field is at the moment, and there's, in terms of evoked inhibitory input to these cells, Hendrik Jorntel has done some work. So this is in decerebrate cats where they actually remove the cortex, so it's not under anesthesia, and then they're using sensory stimulation or electrical stimulation of the forepaw to give sensory input to the cerebellum. And all that they could find, I mean, these are not the best recordings, but they could see that upon stimulation, you could see a sort of hyperpolarization of the membrane potential. And if you look at the average, there's an indication that you get a sort of slow inhibitory hyperpolarizing effect after stimulation. And depending on the membrane potential and the reversal potential of chloride, it would fit with the fact that this is GABA receptor mediated, but it's really loose. It's not very well defined. And that's effectively where the field is in terms of understanding what type of inhibition granule cells actually receive in vivo and how that inhibition is actually recruited, which is why we embarked on the study to really try and separate out and decipher why is there three modes of inhibition and what do those three modes of inhibition actually do for sensory information processing. But we'll cover that on Thursday's lecture.
Is the circuitry primarily feed-forward in the cerebellum?
Yes, it's a very feed-forward system. When in the cerebellar cortex, yes. So it just basically comes into the granule cell layer, up into the molecular layer, cross Purkinje cells, and then out.
No feedback at all?
So there's feedback in the lower center, so you've got sort of feed-forward feedback in the DCN4. So where the cerebellum outputs to is the deep cerebellar nuclear, also gets copies of what is going into the cortex. So the complexity and the feedback, the sort of feed-forward, the more complex nature is down in the DCN. But in terms of the cerebellar cortex, it's all just feed-forward. The inhibition is
somewhat restricted.
yes, less so now because actually, again, another decade has gone on and people are now disputing the fact that the feedback inhibition onto Golgi cells it's actually cross-connectivity between Golgi cells. So again, the sort of, if you took a cerebellar enthusiast and told them to draw the circuit, as you're talking about the feed-forward circuit and where there would be feedback loops, there's bit work done by Wade Regeur's lab recently that shows that actually one of that stereotype schematic diagram, we can now remove part of that feedback mechanism. So now it seems as though it really is a feed-forward way of processing information. A lot of that's done in slice work, so again, what we don't know is whether there's recruitment under particular circumstances. So when you go into slice preparation, you may just be artificially stimulating and you never see these feedback loops, but it might be because you haven't recruited the particular cell types in a particular order or fashion that you don't see it, but that's still to be decided. So that's where the field is, and yeah, we'll go on Thursday. But in terms of whether inhibition is important in the cerebellum, in terms of Golgi cell inhibition, this was some work done that hasn't been followed up since, but it was way back in 1998 by Nakanishi's group, who basically set up I think it was mouse, I'll say it's mouse at the moment, where they could actually inject a drug that would specifically ablate Golgi cells. So it was a molecular technique that they set up, that the Golgi cells were receptive to this drug, and the drug would actually kill all the Golgi cells that were there. Then they could look after, so time point being 0, then they could look days after that at whether it affected normal motor coordination and motor learning. And that's what they did here. So here's wild type, and they basically just dip the paws in ink and then look at the gait of the animal. And gait is a good measure of whether you have normal gait, and the gait can be disrupted if you have cerebellar ataxia or if you have disruption to cerebellar function. So if you have to start to have abnormal angles or your footprints go in odd places, then it means that it's disrupting gait and your normal locomotive function is reduced. And that's what they found, that the actual gait of the animal was severely perturbed once they had ablated these Golgi cells. And then if they looked at motor learning, so I don't know if you've all heard of the rotor rod, where it's a sort of slowly rotating rod, which you can choose the speed at which it rotates, and if you place a mouse on it, it will learn over time to actually be able to accommodate the speed, and it will stay on the rod. For the first 2 or 3 days, it will just fall off, excuse me, if it hasn't been trained, but over time it actually learns to be able to balance on the rotor rod, and that's sort of seen as a form of motor learning. So once it's trained in the wild-type mouse, the animal can stay on the rod pretty well for about 2 minutes. But when they inject this drug over successive days, as more and more Golgi cells die out and that inhibitory control over granule cells is lost, then the ability of this mouse to actually stay on the rotor rod at all is lost. So its ability to balance is lost, and its ability to learn how to actually maintain its balance on the rotor rod is lost. So inhibition is pretty important in terms of regulating information in the input layer of the cerebellum. So that's the input to the granule cell layer, how that's actually controlled, and what's the next layer in terms of the excitatory inputs coming in comes in both parallel fibers and that single climbing fiber, or the olivocerebellar pathway, is what takes information into the Purkinje cells. So parallel fibers or granule cells have an ascending axon. So here's a picture here. There's a single-filled granule cell from Stefan Brenowitz in Wade Regeur's lab, where it's filled with a calcium dye. It doesn't really matter, but it shows you the morphology, where here's a granule cell as an ascending axon, which usually ascends up through the granule cell layer. But you can see that there are these hot spots, or the little white bits you can see here, are actual axon varicosities, which are presynaptic release sites. So even on the ascending axon, you will get release sites, which is thought to activate directly the Purkinje cell, which is directly above that granule cell. Then the ascending axon bifurcates, and as we talked about before, you get parallel fibers, and you can see the beaded nature of these parallel fibers, where each of these are release sites, and they course through the Purkinje cell and produce a single synapse with a spine that's present on the Purkinje cell dendrites. As I said, there's about 200,000 different inputs, and the parallel fibers themselves are 2 millimeters in length. So, and this is, I guess, one of the real big questions I think that people are starting to try and answer now is, if you have, we've talked about the mossy fiber comes in, activates 20 to 50 granule cells. So let's say there's a sub-cluster of granule cells that are excited. They send information up, potentially can affect the Purkinje cells which are above them. But what is really not known is if we activate the cluster of granule cells that are here, where do they actually make synapses? Or where do their parallel fibers actually make synapses? Because it could be on the Purkinje cell which is here, but if this is running for 2 millimeters long, it could be a Purkinje cell that's across here. It could be a Purkinje cell that's this way because it's bifurcating and going both directions. So again, when we're talking about the complexity of understanding what the cerebellum does, how the hell do you organize that? You've got 1 billion granule cells. Each of those are release sites. It courses through the entire dendritic tree of each of the Purkinje cells as it goes along, and we have no idea where they're actually making their synapses. So how do you work out, even if we knew what information is coming in through that mossy fiber, we don't actually know what that means in terms of which Purkinje cells are actually going to get that information to integrate it along with other information to produce some form of meaningful spike output. So I don't know, are you aware of Ed Calloway's method of single synapse presynaptic labeling of all the presynaptic partners? Have you heard about this? Okay, and in short, it's basically a method you can, using viruses, infect one cell. So let's say it was a Purkinje cell. You infect one cell, and this virus jumps back and transfects or invades all the presynaptic partners to that cell. So if you, and it can express, like, say, GFP or a fluorescent protein. So that then what you can say is, and it's a beautiful method, because then if you can infect that one Purkinje cell, then you can say, okay, where are all the presynaptic partners to that Purkinje cell? And then if we could get that just from an anatomical point of view, then you could take long slices through the cerebellum and go, okay, this Purkinje cell gets a subcluster of granule cells here, across here, and across there. And then with other additional genetic mapping tools, then you could map what inputs those granule cells actually get. Then we can start to build the picture of how the anatomical organization is and what information is actually conveyed along those different fibers. But we are, at present, far from that goal. And I think it's probably the holy grail of really trying to understand how the cerebellum actually works. So there's a few people going after it, but it's technically super challenging. Parallel fibers, what do they actually do? I think Purkinje cells are, they're a beautiful cell in terms of the dendritic arbor, but you can see from P15 up to whatever that is, P90, the sort of complexity of the dendritic arbor of the Purkinje cell. As I say, it gets about 200,000 different inputs. They're each excitatory, but the sort of power of the parallel fiber is rather small. Each EPSP, or excitatory postsynaptic potential, that you get is relatively small. So it's all about the integration of many different inputs to really affect, let's say, one dendrite or the spiking nature of that Purkinje cell. So you usually get clustered activation of parallel fiber inputs to really get, and down here is a calcium imaging. So this is clustered activation of a number of parallel fibers coming in, then it will change the calcium levels within that particular dendrite. And then it's the coordinated activity of many different clusters of parallel fibers activating different dendrites that will then feed in and change the spike output pattern of the Purkinje cell. But Purkinje cells are spontaneously spiking, so if there's no synaptic input, they spike at about 30 to 50 hertz. But parallel fibers can obviously give a little kick of excitation and can increase the frequency or the timing of these simple spikes that occur within the Purkinje cell. So parallel fibers generally give a little kick of excitation. It's the integrative effect of many different parallel fibers that will actually really change the spike output pattern that's going to descend out of the cerebellar cortex. Climbing fibers are very different. We mentioned at the start that an adult Purkinje cell only receives one climbing fiber, but as I said, it wraps or ensheaths the proximal dendritic tree of a Purkinje cell. And this is work done by Matthew Zuber-Friedman, again in Wade Reger's lab, where he's reconstructed. In gray is the proximal dendrite of the Purkinje cell. In blue is the climbing fiber axon. And in red is each of the release sites that are present on that single axon. So I think you can see from here that even though it's a single climbing fiber, there are release sites studded all the way along the whole of the climbing fiber. And when an action potential invades in here, the probability of release is pretty high at these synapses. So you get a rather large release of transmitter across the entire proximal dendrite. What that means in terms of what you record, and this is in voltage clamp from a slice recording, where we're looking at excitatory postsynaptic current. But if you stimulate electrically the climbing fiber coming in, I think you can see from the scale here, the size of that input is enormous. I mean, a parallel fiber input would be in the order of about 10 to 20 picoamps. And a single climbing fiber input is it's an order, 2 orders of magnitude larger than that, and you get, 1, 2, and people have seen up to 4 or 5 nanoamps of current coming in. So it's a huge all or nothing input that the Purkinje cell receives, which keys into, in terms of if it's an instructing or a teaching signal, you can get a better activation in terms of if these receptors are going to cause excitation and cause increase in calcium, then by hitting it with a huge input, you're going to get a huge amount of calcium entry, and therefore can start to induce the plasticity mechanisms that we were talking about that could be important for cerebellar learning. This all or nothing input produces in current clamp what is classed as a complex spike, which is an initial fast sodium spike, and then these spikelets that ride on top of a sort of prolonged depolarization. And this is what the Purkinje cell sees, so this complex spike. But it's a little more complicated, and work done by Alex Matthey in Mike Heuser's lab. We did some recordings from inferior olive cells, which inferior olive cell axons are climbing fibers. They're what project up into the cerebellar cortex. But he did some recordings where he did whole cell recordings in slice, whole cell recordings from inferior olive neurons, and then because these axons are usually cut in a slice preparation, he could record from a blebbed end with another electrode. So you could record what spikes were leaving the soma, and then record the spikes that were actually being transmitted down the axon, or faithfully being transmitted down the axon. And that's what you can see here. Here's a somatic recording, and what he found in olivary cells is you get this initial sodium spike, and then there's this prolonged depolarization, and you get these little spikelets. They don't look like very much when you're recording from the soma, but if you go to the axonal recording, which is maybe down here, actually each of these little bumps are faithfully propagated as a spike. So a single action potential on olive cell actually results in a burst of climbing fiber inputs that will propagate up into the cerebellar cortex. So what does that mean in terms of what the climbing fiber sees when it's up here? So this was work done by Sarah Ho, again with Alex Matthey in Mike Heuser's lab. Is what they did was they went in vivo, under anesthesia, patched Purkinje cells in relatively young animals in voltage clamp to see whether they could see any indication that actually if the climbing fiber or there's trains of action potentials coming down the climbing fiber, you would expect to see bursts or trains of excitatory postsynaptic currents in the Purkinje cell. And that's kind of what they saw. And what they used was sensory stimulation. So they were giving ear puff, which would actually trigger climbing fiber activation. What they saw was, in some cases, you would just get a single EPSC. So it's as if the input from the olive cell is only just giving one spike. But then also in other trials, what they saw is that they could see doublets, triplets, and in some cases 4 inputs, suggesting that in actual fact those the train of action potentials that's leaving the olivary cell actually arise at the Purkinje cell to produce a burst of EPSCs. And as I said before, a single climbing fiber stimulation can produce this rather large complex spike if you're looking in current clamp. So what does this mean in terms of if you get a burst of inputs, what does it mean for the complex spike that we see in the Purkinje cell? And what they found was that depending on the number, so this is back in slice again, but they stimulate the climbing fiber once, you get this complex spike with a single fast sodium spike with a few spikelets. But if you stimulate the climbing fiber as if it was a train of climbing fiber inputs coming from the olive, then what you see is a pronounced elongation of this complex spike with many more little spikelets. They went on to show that actually this is really important in terms of plasticity induction. So getting back to your question before, what they found was that if you get this kind of climbing fiber input, the potential or the probability of inducing plasticity was far higher. So actually, there's even more information. It's not just a case of climbing fiber on or off. There's information embedded within the sort of activity of the climbing fiber inputs that's coming in, which will depict the types and probability of induction of plasticity at the level of the climbing fiber, the level of the Purkinje cell. So at every level, it's a little more complex than just a simple feed-forward circuit. There's at each different synapse and at each different point in the circuit, there's a bit of computation that's being done that will change either the information processing or the level of plasticity that's actually being induced. So, an interesting debate that's been around for quite a while. We're running over. Okay, let's go. We can, the end bit we can skip over a little bit. There's an interesting debate that's been going on in the field for quite some time is, do climbing fibers actually have another role? And there's some work done previously by Yossi Roman from Mike Heuser on whether climbing fibers, well, firstly, whether Purkinje cells are actually bistable. What I mean by bistable is on an up state, they fire lots of spikes, then they switch to a down state where they're silent. They don't fire any spikes at all. Then they switch back up to an up state, fire spikes, and then switch back down to a down state. And then there was an indication from both anesthetized rats and awake cats that actually climbing fibers would, to a certain extent, trigger an up state to a down state or a down state to an up state. So actually, climbing fibers were toggling the membrane potential to allow trains of action potentials to leave the cerebellar cortex or making it silent. And this has been going back and forth for a while, so we had a look at this. And a lot of the work, I'm not going to talk about this because it's an ongoing project that we're doing at the moment, but we're doing a lot of recordings in awake behaving animals. So just doing patch clamp electrophysiology in awake behaving animals from the cerebellum. I'm just going to talk about the motor cortex work at 5 o'clock on Thursday. But we did whole cell recordings from Purkinje cells, so you can see the simple spikes, which are just the sodium spikes here, and in red is where you can see the complex spikes from the climbing fiber input. But what we found, and we've done, I think, 40, 50 recordings now from Purkinje cells, that climbing fibers, well, firstly, the membrane potential is not bistable, or we can't see it being bistable. It's unimodal, so there doesn't seem to be an up state and a down state. And because there's no up and down state in terms of the climbing fiber input, it doesn't appear to toggle the membrane potential in any particular way. So it definitely doesn't switch transitional states from up to down. There's another project we've got going on in looking to how complex spikes actually change simple spike firing, but that's a different story. But in terms of the bistability, I'm not sure if Purkinje cells are bistable, and I definitely don't think that climbing fibers are necessarily toggling the membrane potential in the way it's been thought before. So that's the excitation that's coming into Purkinje cells. But it has to be obviously modulated by inhibition, otherwise you get runaway excitation. So another sort of pet love of mine for quite a number of years, which is unfortunately the project I'm not going to talk about, is we're doing a lot of recording, in vivo recordings from molecular engineers to try and understand what their function is in terms of regulating Purkinje cell spike output. But going back to some beautiful illustration by Pillay and Chan Pillay, there's two different types of interneurons in the molecular layer. So these are the ones that actually feed into the Purkinje cells. You get stellate cells, which are in the outer third of the molecular layer. They're relatively confined in terms of their dendritic structure, and then send out a long axon which can course along the sagittal plane of the cerebellum. And then you get basket cells, which are really down in the Purkinje cell layer. So these are out close to where the dendrites are, and the basket cells are right down where the soma is and the axon initial segment is. And these form if you can see them here, these little basket-like structures which really envelop the Purkinje cell, and they produce really strong inhibitory input to the axon initial segment where the Purkinje cell will generate its spiking. So it can have a profound impact on the ability of the Purkinje cell to actually evoke spikes, or it can at least have a profound impact on suppressing the spikes that the Purkinje cell is intrinsically generating. So just a little snapshot of what some of the results that we're getting, and an interesting feature, we're sort of on the last experiments now to be able to wrap up a paper. But interestingly, you've got hundreds of molecular layer interneurons. And we've patched on to 20, 30, 40 of these cells. And what we find is when the animal actually starts to initiate voluntary locomotion, so it's just running on a 24 centimeter polystyrene ball that's friction reduced, so if it wants to run, there's no friction or load on it. It can run, it can walk, and it can groom. But what we find is the interneurons are spiking, probably on average about, let's say, 10, 20 hertz. But when the animal initiates movement, every single interneuron that we've ever recorded from cell attached or whole cell increases its firing rate during movement. So it's not as if there's subpopulations of cells that don't respond during locomotion, some that do. All the cells that we've recorded from actually increase their firing rate, which will send GABAergic projections onto interneurons. So the main question that we're going after, and the question that we're trying to resolve, is what is the actual role of interneurons in terms of regulating Purkinje cell spike output?
So do these interneurons receive input from just the mossy fibers?
No. Again, another debate. So the majority of their input they're definitely getting is the mossy fiber granule cell parallel fiber input. That's their main drive. But there's been in vitro papers and in vivo papers that show that climbing fibers can also stimulate interneurons. So they're almost sensing the large excitatory input that the Purkinje cell is getting to modulate its activity. It's less well documented, but there's definitely enough evidence out there to suggest that interneurons get climbing fiber input. So they could be sampling the same sort of inputs as the Purkinje cell is getting. But why might interneurons be important? Well, I said before that if you take away all synaptic input to Purkinje cells, they're intrinsically firing at about 30 to 50 hertz. So they're constantly putting a tone out. And interestingly, Purkinje cells are GABAergic. So if there's a tone of 30 to 50 hertz constantly, it's constantly releasing GABA and inhibiting the deep cerebellar nuclei. So there's no information, let's say, going out to downstream motor centers if you're inhibiting these neurons. But if you stop the spiking of Purkinje cells, then you release this inhibition. You allow the deep cerebellar nuclei to then spike, which will then send information out to potentially modulate downstream motor centers or the downstream motor action. So it's our belief that actually interneurons give you a real enhanced computational significance. In fact, if you can regulate or suppress the intrinsic spiking of the Purkinje cell, then when you silence, these periods of silencing are actually really important to send out packets of information, because when this is silent, there's packets of information being sent out from the DCN. This is spiking. Then it'll go back to potentially being suppressed here. Then information in the DCN will go out when you relieve the inhibition down here. And this could be a way in which you'll then get patterns of activation and inhibition of downstream thalamus and descending motor tracts, which can actually fine sculpt the motor movements. So we're really interested in getting at how important is, what function do these interneurons serve, and how important are these sort of pauses or gaps in Purkinje cell firing in terms of what information actually leaves the cerebellum. We're doing all this in awake behaving animals to really get at the unperturbed system. And the best indication that we've had in terms of the interneurons really are quite important is from work done by Chris Dezou, who's a sort of pioneer in the field. And with Pierre Wolf, who's now in Germany, what they did was they generated a mouse that the Purkinje cells had GABA receptors that were not functional. So effectively silencing the GABA receptors on the Purkinje cell. And what this will allow, so here's some voltage clamp recordings from a slice preparation, I think, looking at in voltage clamp. So each of these inward currents are actually inhibitory postsynaptic currents. So these are iPSCs. In control, this is a normal wild-type mouse. And then, well, actually, they actually use it that they can apply a drug to silence the GABAergic input. So what they do is in control, here's your recordings, and then they can silence, by applying a drug, they can silence the inhibitory input to these Purkinje cells. So these Purkinje cells now receive no inhibition whatsoever. And to cut a long story short, why it highlights the importance of interneurons, when they look at, they're basically looking at vestibular ocular reflex, but it doesn't really matter. They're looking at motor learning in mice. And what they found was that in wild type or control animals, the consolidation of that memory, so when they trained on day 1 and then left them overnight and trained on day 2, the ability of the mouse to remember the motor task it was performing in day 1 is fine. It's about 80% on wild type mouse. But if you knock out the function of interneurons or knock out this inhibitory control of Purkinje cells, then their ability to retain or consolidate that information from day to day is completely lost. So in terms of motor learning, it seems that interneurons are pretty important. But we've got data to suggest that actually even normal function of the cerebellum is highly, highly important, that you've got regulated activity of interneurons.
You would look as if it would be just delayed, and I mean, what happens after these days? Could they eventually go to the normal?
It looks as though it's heading that way. Potentially, yeah, just repetition might put it back. They didn't go much beyond that. But potentially, yes. Also, even though it's drug-induced, or even if you use a transgenic animal that's got a knockout of a particular receptor, you've got to be worried that actually every brain area has an ability to upregulate other types of receptors to be able to compensate to a certain extent. So whether this is truly, if you could go in, take a snapshot, what happens there, put it back on again, put it back off, so on and so forth, then you might get a different, we don't know whether you get a different answer or not. And so it could be a caveat that, but it at least is a good indication that an intact system with inhibition is important for motor learning. So I think it's probably safe to stop at this point. It's just a very, very brief overview of a couple of things about cerebellar-dependent motor learning. But if you're interested in that, there's a plethora of papers out there you can go and look at. So in terms of what you need to know, which will give you the background for what we're going to talk about on Thursday morning, and then we'll leave it there.
Any further questions?
Yeah.
So thank you. We'll meet on Thursday morning again. I will send the papers for the spinal cord presentation today with some delays in faster. It will certainly be today.
Will you send the new syllabus?
Yes, I will, together with that, I will send.
Is there a discussion paper for next week? Or is it
There
just
will be another one, together with these ones, I will send also the book with them. So it's two presentation and one discussion.
also
For
for
spinal cord.
cerebellum intuitive?
Cerebellum is not secret.
Right.
So I think
So on the 10th we have cerebellum papers. Is there a discussion?
yeah, I will send them as well.
Do you want these slides?
Yes, it doesn't have to be now. Well, anyway, so I'm very happy to go ahead.
Okay, so when is the next result of the paper?
Yeah, thanks.
three core mammals. We've done. We started three, and we're about maybe
הוא אמר
75%
שזה הביוב, הוא
done.
אמר שהביוב
But the
סתום,
problem is
צריך
we
לשטוף
like
את הביוב.
we have to write functions. We wrote the functions, but
הוא לא אמר
we
שהוא
don't
יעשה
think
את זה
our
בלי
results are
הצעת החוק.
correct because we have
היא לא
a
יכולה לעשות,
mistake. We're not
יש לה בעיה, היא לא
we wrote
יכולה
something. We're not sure if it makes sense. Like
על
because
חשבון
we do
אחד בלי לקבל אישור.
a
לא נתנו לו אישור, ואני בפירוש לא נתתי לו אישור, הוא גם אמר לי שיזכיר אותו, כי הוא עוזר לנו לפעמים, הוא אמר לי, גם צריך לעזור לנו,
we're on a meeting with Ron today
הוא אמר,
after the
אני נראה בן אדם נחמד, אמרתי, תשמע, אני לא יודע איך אנחנו נסגור את זה, אבל כשחזרתי בערב, אבא שלי היה לו פקס, בדיוק יצא, עצרתי לי את זה, פתחתי את הכל, לא דיברתי, אמרתי, לא, אני לא יכול לפתור את זה, בכל מקרה, אני דיברתי עם מירי, ונשלח לה הצעת מחיר בבוקר, ואז נראה מה עושה. אז אני לא יודע אם הוא בכלל עשה את זה. לדעתי הוא לא עשה את זה. זהו, זה לא בדיוק חינוך שלו, זה חינוך שהוא מלמעלה.
So this one is basically the same thing as the example we made in class. In class we saw a bounded function, 0 and math. We have to look at a output rate that's half bounded, so it's 0 and so this is things we did in class, just a little. This I don't remember what this is. No, I kind of remember. So this is we have this network here. X input. They go to one output over here, and then they go to the dimension is condensed and then expanded again. And we have to show optimality when it makes sense that U and W should do the same thing. There's a proof. I don't remember. We've moved on. This is tough. So we spent a long time trying to understand this equation and this equation. And then we spent a long time trying to write the function. So the first thing is we have to write this function. Right, if you're given a vector pk, constant mu, constant epsilon, constant delta, you can express this function in that way. Okay. The second thing you do is then you have to express this function in that way. Where you do this summation of this, and this is vector summation. Yeah. And you have to express all of this without the equal to 0. Then you use this function to do you know what f-solve is?
לא, but
F-solve is, you have this equation, there's all these variables in it. So F-solve says, if I tell you the value of, let's take this example. If I tell you the value of pk and epsilon and delta, you can find mu